All right. So in the next section, we are going to look how scientists can determine whether a population, okay, is evolving or not. So far we discussed the mechanisms of genetic variation, and genetic variation might lead to the evolution, or it's a must in order for a population to evolve. So evolution of a population cannot happen if there's no genetic variation. Okay. So when we talk about evolution of population, we're talking about changes in their phenotypic variations, and phenotypic variations requires genetic variation. So if there's no genetic variation happening, if there's no gene variability or DNA variability happening, there will be no genetic variation. If there's no genetic variation, there is no phenotypic variation. If there's no phenotypic variation, this means there is no evolution within the population. Okay. Now, but you have to understand, if genetic variation happens, that does not mean that the population must evolve. So genetic variation does not guarantee that evolution at the level of population will happen. And we could determine if a population is evolving or not by applying what we call the Hardy-Weinberg equation, and this is what we're going to learn in this section. So before we start discussing this equation, the Hardy-Weinberg equation that will tell us whether a population is evolving or not, let's first define a population. So a population here is a group of individuals, a localized group of individuals capable of inbreeding. Inbreeding means mating and producing fertile offspring. So this is very important. Inbreeding and producing fertile offspring. Okay, because sometimes like in animals, some different individuals, they can mate, but they cannot produce fertile offspring. If you, for example, if you're talking about horses mating with donkeys, they will produce new offspring, but it's not going to be fertile. Okay, here we're talking about individuals of the same kind or the same species that are able to mate together to produce fertile offspring. Okay, fertile means خصبة. Inbreeding means تزاوج. Now, these populations, they might be found in a geographically restricted area. Let's say that we are looking at an individual of birds on an island. Okay, maybe there is an island, and we're looking at certain types of birds on an island. Or maybe the population might not be. So this is, here we're talking about isolated population. Isolated means the individuals of these populations, they are not able to mate with other populations found elsewhere because they are found in an island, isolated geographically. Okay, isolated معزول جغرافيا. Okay, but sometimes you have, let's say, two populations, or sometimes these some populations are not always geographically isolated, and you might have, let's say, two populations of one kind of animals, let's say in your book we have I don't know if we have, yes, so here we have two populations, let's say, of animals, and these two populations, the 40-mile herd and the porcupine herd, they are not geographically isolated, although they occupy, let's say, uh, um, occupy, very close by areas, but they also overlap in the area that they can live at. So as you can see, this map shows the basically the range or the area in which this porcupine herd lives, and here we have the area in which the 40-mile herd live, and both, or members of both population, if they are these animals are, let's say, compatible, they can mate in this area, they can meet each other and inbreed, and maybe they could produce fertile individuals. So here we're talking about two populations that are not geographically isolated, and they can interact with each other. Okay. Another term I would like to define here is the gene pool. Okay, so gene pool is basically, if we look at the at a population and we determine all the alleles for all loci. Okay, here loci includes genes and some DNA sequences that are not genes. So all loci in a population. And we could say that if there is only, let's say, one allele for a particular locus, okay, and all individuals in a population, they are homozygous for that locus, because all of them, they have the same allele, then that allele is said to be fixed, okay, in the gene pool. Okay, so if there's one locus that has only one allele, and this allele is found in all loci inside individuals, all individuals of a population, that locus or that allele is said to be fixed. Okay. Of course, some other loci or genes, they might have one or more, two or more alleles. And if we're talking about diploid organisms, they could be either homozygous or heterozygous for that loci. Okay. Now, for example, if you remember when you learned about this wild flower that shows incomplete dominance. So here we have an example of incomplete dominance, where you have two alleles, the CR allele and the CW allele. Okay, so the color of this wild flower is determined by, let's say, one gene locus, and there are two alleles of this gene locus. Okay. So every gene locus still have gene pool? No, so the gene pool is basically all alleles for all genes. All alleles, the gene pool consists of all alleles for all genes. Of course, some genes will have only one allele, some genes they will have two alleles, some genes three alleles. If you're talking about the ABO system, we have many alleles. Okay. So what is it exactly? It's not a thing. It's just a concept. Okay, you basically analyze all the different alleles for different genes within our population, and these alleles will represent the gene pool. Okay, so the idea of the gene pool will become more clear as we discuss the Hardy-Weinberg equation. So here we're talking about one gene locus responsible for the flower color in this type of plant. Okay, and the genotype could be homozygous for the R allele, homozygous for the white allele, or heterozygous. And here we have each genotype reproduces a different phenotype. Okay, so here we have incomplete dominance. So the two alleles, we cannot establish the dominance. Each one of them is not dominant over the other. We have incomplete dominance. Now, we can calculate the frequency of each allele. So here we have two alleles. We can calculate the frequency of each alleles. Of course, plants are diploid. If the plant is diploid organism, this means that we have two loci or two copies of the gene. The two copies could be identical, like RR, identical WW, or different. Okay? So if we, for example, if we're talking about, if you analyzed 500 plants of this type of wild flower plants, okay? The 500 plants, they will have how many alleles that are important for determining their flower color? Yes, locus, loci, locus. Okay, loci جمع, locus مفرد. So they will have these 500 plants, they will have 1,000 alleles that are important for determining the flower color. Okay. If for example, I told you that there is 320 plants with red flower color, 160 pink flower color, and 20 white flower color. Okay, so you have 500 plants with these numbers of different plants with these different phenotypes. So you can basically calculate the frequency of these alleles. How? So the 320 plants, we know that red flower color is produced when you have when the plant is homozygous for the R allele. So you have 320 multiplied by 2. This means we have 640 CR alleles. The pink is basically produced by the heterozygous genotype. So you will have 160. So heterozygous plants, so we have 160 pink, which means half of these pink plants, they will have 3,200 alleles. Half of these 160 are CR. The other half 160 will be CW. So 160 multiplied by 1 is 160. Also CR this is from the red plant, this is from the pink plant. Okay, so now if you combine these, you will get what? You will get 800 alleles or 800, let's say, CR alleles. Out of how many alleles? Out of 1000. So if you divide 800 over 1000, you will get 80%. So this is the allele, the CR allele frequency. Typically, the 80% is, or if you divide this number over this number, it's not 80%, it's 0.8%, 0.8. So this is the allele frequency. The allele frequency for the CR allele. For the CW, the CW allele frequency will be 0.2. So the allele frequency of CW and CR should basically equal to 1. So allele frequency of CR plus CW should equal to 1. If you have more than one allele also, the allele frequencies of all alleles for one loci should be equal to 1. Okay? So if there is a loci that could be basically could be one of two alleles, and we designate P small letter and Q small letter for either one of these allele frequencies, the frequencies of both alleles in a population should add up to 1. And this applies also if there is more than two alleles. So if there is a loci that could be one of three alleles, the allele frequencies of these three alleles should also add up to 1. The sum of all allele frequencies should always add up to 1. All right, so let me just, so we still have, okay. So the Hardy-Weinberg equation is used to describe the genetic makeup we expect for a population that is not evolving at a particular locus. So here we're talking about a particular locus or about a particular, let's say, gene. Okay. Yes. So yes. So the W, so the W is calculated like this. You have 20 flowers that are white. So 20 by 2, 40, plus 160 from, so this is the W, you have these two, 40, plus 160 from the heterozygous, you get 200. So 200 from 1,000 plants, from 1,000 alleles is 20%. So 200 over 1,000 alleles equals 1.2. We did calculate them here. 20 white flowers, they will contain 40 CW alleles. And 160 CW alleles from the heterozygous, 160, 40, 200, 204 over 1,000.1. All right, so scientists uses this equation to determine whether a population is evolving or not, by comparing the observed genetic makeup of the population. Of course, this is done at one locus at a time. So we use this equation to look at a particular locus, a particular gene, a particular alleles of genes, of a gene, okay? By looking at the allele frequencies of a particular gene, of one gene at a time. So basically, we're comparing the real genetic makeup of a particular gene with the expected genetic makeup of a particular gene. If both the real genetic makeup of a particular gene differs from the expected genetic makeup of this same particular gene, then the population might be evolving. Okay? And this is what we are going to learn about, inshallah, in the next lecture. So here we have, if there is a population that is randomly mating and we have Mendelian inheritance occurring, if the alleles and genotype frequencies remain constant from one generation to the next, then this is basically a population that is hardy, that is in hardy-Weinberg equilibrium. Okay, and the population is not evolving. Okay, but if the allele and genotype frequencies are changes from one generation to the next, the population will not be a Hardy-Weinberg will not be in Hardy-Weinberg equilibrium, and the population is evolving. دكتور، طب شو بنستفيد لما نحسب نسبة كل الأليل؟ Yes, so when we calculate the allele frequencies, this is what we're going to learn in the next lecture. Calculating the allele frequencies will tell us whether using the Hardy-Weinberg equilibrium will tell us whether the population is in Hardy-Weinberg equilibrium and it's not evolving, or the population is not in Hardy-Weinberg equilibrium and it is evolving. So the Hardy-Weinberg equilibrium, just a quick reminder, or equation, can give us an idea about whether a population is evolving or not. More specifically, it will tell us whether the allele frequencies for one or a number of loci is changing from one generation to the next. So the Hardy-Weinberg allows us to compare the allele frequency, the expected allele frequency, with the observed allele frequencies. So we can expect allele frequencies for a population, and we can observe or determine the actual allele frequencies for a population and compare these allele frequencies. If they are different, this means the population maybe is evolving. Now, a population where the gametes that are contributing to the next generation, they are produced randomly, and we have Mendelian inheritance occurring in this population. This means that if the alleles and the genotypes frequencies are constant from one generation to the other, this means that the population is in Hardy-Weinberg equilibrium and the population is not evolving. So the Hardy-Weinberg equilibrium describes the constant frequency of alleles in a gene pool. Remember, the gene pool contains all the alleles within a population. For example, if we have 500 wildflowers, okay, these wildflowers, they have two alleles for the loci determining the flower color. We have the CR allele, the CW allele. 500 flowers will contain 1,000 alleles, because each wildflowers are diploid organisms, which means each gene, there is 2 copies of the gene responsible for determining the flower color in the wildflowers. So the allele frequencies, based on our calculations in the previous lecture, for the CR is 0.8 or 80%, and for the CW is 0.2. So imagine if all alleles for a given locus. Here, the locus used to determine the flower color in wildflowers. Imagine if all alleles are placed in a large box or a large bin. So basically, this bin or this box holds all the alleles, all the population, let's say, gene pool for that locus, for that gene. Now, in order for reproduction to happen in this population, male gametes or sperms should be selected, okay, randomly from this box, and eggs should be selected. Okay, these alleles will be found in order for reproduction to happen. Imagine these are gametes, male and female gametes, and they are color-coded according to the allele, according to the flower allele that they contain. So you could think of this as a, let's say, a gamete. It could be a male or female gamete, male or female gamete with a CW, okay, and this one a male or female gamete with CR, okay. Of course, if you're talking about a population consisting of thousands of or 1,500, let's say, wildflowers, like in our example, we know that there will be 800 in this box, 800 gametes with the CR and 200 gametes with the CW. Now imagine these are the gametes that are found in the population. In order for reproduction to happen, random selection of, let's say, sperms or male gametes and eggs, female gametes. Now, because of the allele frequencies that we already determined, that are already known for this population, the chance that a sperm will have a CR allele is 80%, because 80% is basically the chance of having the CR allele in all the alleles within the population. 20% chance that the sperm will have the white allele. Same thing applies to the female gametes or the eggs. Okay, so these are all the alleles found in the population, and these are the percentage or the probabilities of having these gametes if a gamete is chosen from, if female or male gametes are chosen from the population in order for mating. So now we can calculate the frequency of genotypes based on the frequencies or the probabilities of these gametes. So the probability of mating between a sperm, okay, with the CR allele and an egg with the CR, also with the CR allele. So here we have P2. So we use the multiplication rule. So 0.8 is 0.64 for this genotype. Okay. Same thing applies if a white allele gamete or a male gamete with a white allele fertilizes an egg with a white allele. So we have, so we use the multiplication rule. So Q squared, 0.2 multiplied by 0.2, 0.04. Now for the production of offspring with the heterozygous genotype. Heterozygous genotype could happen by two ways. A male gamete with the CW and a female gamete with CR. This will be to produce CW-CR, or a male gamete with CR and a female gamete with CW. This will produce also CR-CW, which is the same. Okay, so we multiply this. We use the multiplication rule to calculate the frequency of the genotype resulting from a CW containing gametes and a CRW containing gametes. Same thing applies. Then we use the multiplication rule to determine the final probability of having let's say, having this genotype. So CW is 0.2 multiplied by CR, which is 0.8, 0.8, 0.2. So here we have 0.16. 0.16, we use the addition rule to determine the final genotype frequency, which is 0.32. Or you just multiply the probability of the first allele, the allele frequency, you multiply it with the other allele frequency, and then you multiply by 2. Okay, this is the straight way. This is just an explanation why we got this number. So now, these are the genotype frequencies, okay, not the allele frequencies. So these are the allele frequencies. Remember, this one and this one, these are the allele frequencies. Okay, and the ones in the red, the blue, these are genotype frequencies. And notice that 0.64 plus 0.3 for this population plus 0.04, okay, adds to 1. Okay, same thing, just a quick reminder, 0.2 plus 0.8 allele frequencies adds to 1. So we have this. So this is what we have done, described using the Punnett square. So you learned about the Punnett square in the Biology 101 course. Here, the same thing. We could use the Punnett square to determine basically the genotype frequencies. Okay. So again, the probability of having a sperm with a CR is 0.8. A sperm with the CW is 0.2. Same thing applies to eggs, and we already got this. So we already explained this, and we already calculated the percentage of the or the frequency, the genotypes within in the next generation of a population consisting of 500 wildflowers. So the idea here is that if these 500 flowers, they reproduce to produce the next offspring or the next generation, these are the expected genotype frequencies in the offspring or in the next generation. This is very important. In the next generation. Of course, if the mating between these let's say these plants is random. Okay, this is very important. So let's have a look at the Hardy-Weinberg equation. So if the P and the Q represent the relative frequencies of the only two possible alleles in the population. Okay? So here in our example, the P is basically corresponds to the CR and the Q corresponds to the CW. Then this equation, this is the Hardy-Weinberg equation. The allele frequency squared of one allele plus 2 multiplied by the allele frequency of the first allele and multiplied by the allele frequency of the second allele plus the square of the allele frequency of the second allele, these values should add up to 1. Okay? So the P squared, the Q squared, they represent the frequencies of the homozygous genotypes. In our example, the CR, 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 C, W. And the two PQ represent the heterozygous, the CR, C, W. So this is the Hardy-Weinberg equation. It is used to calculate the expected frequency of genotypes in the next generation, in the offspring of a population that is randomly mated. Okay? So again, this equation is used to calculate the expected genotype frequencies in the next generation if the population is mating randomly. Okay? Now, in order for a population to be in Hardy-Weinberg equilibrium, there are a number of conditions that should apply to this population. So the Hardy-Weinberg equation could be used to describe a population that is not evolving, so that the allele frequency and genotype, the allele frequencies are not changing from one generation to the next. Okay? So allele and genotype frequency, they do not change over time or from one generation to the next. Now, if a population is not in Hardy-Weinberg equilibrium, this means that some of the conditions for Hardy-Weinberg equilibrium are being violated or are not being met. So there are conditions that should be met in order for a population to be in Hardy-Weinberg equilibrium, or for a population that is not evolving as determined by the Hardy-Weinberg equilibrium. So these conditions, the 5 conditions, are the conditions for Hardy-Weinberg equilibrium. If these conditions are met by the population, again, the population will be in Hardy-Weinberg equilibrium, and the population is not evolving, which means that allele and genotype frequencies are not changing over time. Okay. Okay. This is the meaning of a population in Hardy-Weinberg equilibrium. It's not evolving. How did we? And this means that the allele and the genotype frequencies are not changing over. Okay. So what are these conditions? The population should not have mutations or new mutations introduced into the population. Okay. So these are the conditions, and if the condition does not hold, these are the consequences. So a Hardy-Weinberg equilibrium, or a population Hardy-Weinberg equilibrium, will have no mutations or no new mutation, because these new mutations that could be introduced into a population, they will change the gene pool, which means a change in the allele frequencies could happen, or the genotype frequencies in the next generation. A population Hardy-Weinberg equilibrium must randomly mating. Okay. If a subsequent of the population or a subset of the populations are mating or inbreeding, okay, this might basically increases the allele frequencies of certain alleles in the next generation. No natural selection should happen for certain alleles within the population. If natural selection happens, this could lead to the selection of certain alleles and the removal of certain alleles from the population. Okay. The population should be a large population in order to be in Hardy-Weinberg equilibrium. If the population is small, this means that we will have allele frequencies fluctuations over time. Okay, this is because of a process called genetic drift. So we will learn about this this process this presentation, inshallah. Also, the Hardy-Weinberg equilibrium population should not basically have a gene flow from other populations. Okay, again, we will discuss how gene flow could occur and what is the key consequences of this process. So in gene flow, new alleles could be introduced into the population, or some alleles from the population could be moving out of the population. Okay. So these are the 5 conditions that should be met in order for a population to be in hard-to-Weinberg equilibrium. If any of these conditions are not met, the population will not be in hard-Weinberg equilibrium, and this might lead to the evolution or change in allele frequencies and genotype frequencies in the over time from one generation to the next. So again, departure from these conditions might lead to an evolutionary change. And you have to understand that this evolutionary change, okay, it's common in nature or in natural population. But also some natural populations might be in hard-Weinberg equilibrium and might not be basically evolving. Okay. So natural population can evolve at some loci while being in hard-Weinberg equilibrium at other loci. So you have to understand that we could apply the Hardy-Weinberg equilibrium at different loci. Some loci might be evolving and might not be in Hardy-Weinberg equilibrium. Some other loci, okay, might not be evolving. So here, loci, again, when we talk about loci, we're talking about gene locus or gene, okay? Genes, okay? So one or more locus gene loci or one gene locus might be in Hardy-Weinberg equilibrium and not evolving. The allele frequencies are not changing for that gene, or the genotype frequencies are not changing for that gene. But maybe the allele frequencies and the genotype frequencies are changing for another gene. So that other gene will not be in Hardy-Weinberg equilibrium. Okay? So you have to understand, so far we have been discussing just only one locus, the locus that is responsible for determining one phenotype or one character. Okay? But we could apply the Hardy-Weinberg equilibrium to other loci within the genome of organisms. So some applications of Hardy-Weinberg equilibrium, some other application is to determine basically the percentage of a population carrying a specific allele. So the main purpose of using the Hardy-Weinberg equilibrium equation is to test whether a population is evolving or not, is to test whether the allele frequencies and genotype frequencies are changing over time or not. But also another application is to determine the percentage of individuals within a population carrying a specific allele or maybe a specific gene. So let's have a look at an example. So there is a genetic disease called phenylketonuria. Okay. This genetic disease is basically determined by a gene locus in humans. So we have a PKU gene determines whether individuals would have this medical condition called phenylketonuria. Okay. So we know that this gene locus or this locus is in Hardy-Weinberg equilibrium. Why? So before we talk about this, so the phenylketonuria, it's a metabolic disease. It's an inherited disease, okay? And this disease is caused when individuals have a mutant recessive alleles, two two mutant recessive alleles of the PKU gene. Okay? And typically, I think in the United States, because there's like statistics in the United States, it affects 1 out of 10,000 babies born. So to apply the Hardy-Weinberg equation, we have to assume that let's say the United States population is in Hardy-Weinberg equilibrium for the locus, okay, for the locus that is responsible for this inherited disease. So in order for the population to be in Hardy-Weinberg equilibrium for this population, there should be no mutations. And this is the first assumption, that the gene mutation rate in the mutation rate in BKU gene is low, and this is actually true. So no new PKU mutations are being introduced to the population. So this is the first condition of the Hardy-Weinberg equilibrium that is met. Also, we could assume that people, in the United States, they mate randomly. They're not choosing, let's say, specifically their relatives. They are just mating randomly, and this is a valid assumption. We could also assume that there's no natural selection happening because of the different alleles of the PKU gene. Okay, and this is a valid assumption. So you have to understand that natural selection will typically natural selection will select the alleles that will offer a greater chance of survival and reproduction success. So we have a dominant, let's say for this gene, we have a dominant, let's say, normal allele, and we have a recessive, let's call it disease allele. Let's call this, let's call this allele N, and this is like disease D small. So individuals with recessive disease alleles, they will have this metabolic disease. So both alleles, they do not offer survival or reproduction advantage to individuals, which means that there is no natural selection for any of these two alleles happening. We could assume that the population of the United States is large enough. It's extremely large, okay? And then there's no gene flow, because even if there is people coming from outside the United States carrying their own alleles, these people are coming from other populations, and these other populations, they have the same, let's say, ratio of people that are infected. So you have to understand that we're trying to apply the Hardy-Weinberg equilibrium on the population of the United States. Okay, in this population, 1 every 10,000 babies, they have the PKU disease. Okay, so, and you could think of this, the population of the US will have the N and the D alleles, okay? And people from other countries or from other population, okay? Let's say from China, and people coming to the United States from, let's say, the European Union, these are two different populations. There might be some migrations in both directions. This is actually what is meant by gene flow. And here, some alleles, like the N, the D alleles, might be moving from between these populations. Okay, but this will not affect basically the fifth condition of hydrodynamic equilibrium, because these populations might have also the same, let's say, incidence or the same prevalence of people with PKU in, let's say, in China or in Europe. We have the same percentages or the same incidence. Okay, so even if people migrate from China, they will not basically change the allele frequencies of the US population. Same thing applies. Or even if some people from the US, they migrate into China or into Europe, they will not have a large effect on the allele frequencies of the PKU locus, because this population is extremely large. Okay, and the people that are migrating from or into the other populations, these populations have also the same, let's say, prevalence of this disease, okay, or the same allele, let's say, frequencies. Now, so we could assume that the 5 Heidelberg equilibrium conditions are met and they are not violated. We can now determine basically the percentage of people that are heterozygous for this locus. So remember, we have disease allele, and we have the N allele. Assume that we want to know what percentage of people, or let's say US people, United States individual, with the heterozygous genotype, let's say with the genotype ND. Okay, so we could, have, but we know that this disease affects 1 out of 10,000 births. Okay, and so this disease is observed in 1 baby out of 10,000 babies that are born each year, which means that these babies, they have this genotype. Okay, and if we basically have the P as the allele frequency of the N allele and the Q as the allele frequency of the D allele or disease allele. So now the DD, basically the percentage of people with DD genotype, we have to divide 1 over 10,000, which is 0.0001. Okay, so DD is basically D squared, which is basically Q squared. So now Q squared is 0.0001. Q, the allele frequency of the D, which we can know from the genotype frequency of the recessive genotype, is now 0.01. So this is the allele frequency, okay, of the disease allele. We know that from our previous discussion that P plus A plus Q, the allele frequencies of both alleles for the same locus, should equal to 1. So if we know Q, we could know P. So P equals 1 minus Q. So P equals 1 minus Q. We know Q. Q is 0.01, which we were able to determine from the genotype frequency of the recessive genotype, DDD. And now this is the allele frequency of the the normal allele. So this is the allele frequency of the N allele. Now, heterozygous, the allele frequency of the heterozygous could be determined by calculating the 2pq. So this is the 2pq allows us to determine the heterozygous genotype frequency. Okay, so we multiply 2 by the allele frequency of the first allele frequency of the second allele. Now, this is the allele, the genotype frequency of the ND genotype. So approximately 2% of the US population will have the heterozygous genotype. Okay, so this is an example of application, another application of the Hardy-Weinberg equation. Okay. So in the next concept, we're going to discuss more or learn more about natural selection, genetic drift, and gene flow. And these are three processes that could change the allele frequencies over time. Okay. Remember, if we go back, the 5 conditions that that are required for a population to be in Hardy-Weinberg equilibrium. Any deviation from these conditions might lead to an evolutionary change or might lead to a change in the allele, and it might lead to a change in allele and genotype frequencies over time or from one generation to the next. Okay. So let's discuss first natural selection. So the concept of natural selection is basically based on the differential success among individuals in survival and reproduction. So certain alleles will make certain alleles present in certain individuals will make individuals basically more able to reproduce and to survive in their environment. Okay. So within a population, we have a variation in the ability of individuals to survive their environment and reproduce. So some individuals within the population, they have alleles that makes them more suited or more suited to their environment. And these people, individuals that are more suited to their environment, they will be able to produce more. And this means that they will produce more offspring, okay, than the individuals that are not well suited to their environment. Okay. So over time, these individuals that are more suited to their environment, they can survive, they can produce more than the others that are not suited to their environment. So this natural selection, if we're talking about genetically, natural selection will result in certain alleles being passed from one generation into the next. And these alleles that offers advantage in terms of survival and reproduction for the individuals carrying them. Other alleles will be removed from the population going from one generation to the next because these alleles, they do not offer any advantage in terms of survival and reproduction. Actually, these alleles might be harmful to the population, so they're not going to be passed from one generation to the next. Okay. So, for example, in your book, So for example, scientists have found out that after the introduction of DDT, so DDT is an insecticide. So DDT, insecticide. So this is a chemical that is used to kill insects, especially insects that are causing damage to crops or plants. Insecticides. So this insecticide was introduced, let's say, around 1960s. And Drosophila, fruit flies. So before the introduction of DDT, Drosophila flies or fruit flies, they were affected by this chemical. So Drosophila, they might be sensitive to DDT, which means, so fruit flies that are sensitive to DDT will be killed. Some fruit flies became resistant to DDT, and they're not killed by this chemical. So it turned out that these resistant flies, they have an allele of a gene that allows them to become resistant. So we could say that there are some alleles that makes the fruit fly sensitive to DDT, some other alleles that will make these fruit flies resistant. Resistant means مقاومة, and sensitive means حساسة. Okay. So before the introduction of this DDT, okay, scientists found out that the frequency of the resistant allele was actually 0 in the lab. In the fruit flies that are propagated inside labs, not in the nature. But after the introduction of DDT, okay, so we could say that, so here we have DDT introduced. So before DDT introduced, the allele frequency of the resistant allele was 0 in fruit flies. Okay, in the lab, let's say, in labs. And after the introduction of DDT, the percentage of the allele frequency became 37 in fruit flies that are propagated inside labs. So we could say this is the resistance allele frequency. Okay. So after, so this is in 19, let's say, it is mentioned in your book in 1960s. Okay. And we could say that this is 19, let's say, 30s. This allele was not present in the fruit flies that are propagating in the lab. And 30 years later, after the introduction of DDT, after the use of DDT as an insecticide, the allele frequency became 37. So we could say that between, let's say, between 1930 and 1960, okay, the allele, this resistance allele, okay, so this resistance allele might have been introduced into the genomes of Drosophila because of a mutation or maybe it was a normal allele and a mutation changed the normal sensitive allele into a resistance allele or resistant allele. Or maybe it was found in fruit flies, but it was very rare to be found in the fruit flies, and natural selection selected flies that are carrying this allele after the introduction of DDT, and the allele frequency of this resistant allele increased with time as more and more insecticides were used, more and more DDT was used as an insecticide. Okay. So this example suggests that this allele, or the resistance allele, or the allele that confers resistance to DDT, will increase in the frequency in a population that are exposed to this insecticide. Okay, so the exposure, the use of this insecticide to kill flies will lead to natural selection of the flies that are carrying this resistance allele. Okay. So this allele favors the survival of the resistant allele, favors the survival of fruit flies carrying it if these fruit flies are exposed to the DDT. So this is an example of adaptive evolution. So natural selection can cause this adaptive evolution. In this evolution, what happens is that natural selection will select traits that enhances the survival or reproduction increase in frequency over time. Okay, so natural selection basically enhance the ability of fruit flies in this example to resist this insecticide. In terms of genetic drift, this is genetic drift describes how allele frequency could fluctuate unpredictably by chance from one generation to the next. And typically genetic drift will affect small populations. And this genetic drift typically reduces genetic variation because of the random loss of allele. So here we have some events, some random events, okay, might cause the disappearance of certain alleles, which means allele frequencies and genotype frequencies will fluctuate, okay, from one generation to the next. Now, the smaller the population, the likely the more chance that this genetic drift will affect that small population. So we could learn about genetic drift, for example, by looking at this example. Imagine if we have one generation of these wildflowers with these allele frequencies of the CR and CW alleles. Now imagine if something happens let's say something happens to this population, and this event, or let's say maybe an animal came to this population and started eating some of these plants, let's say, or maybe some of these plants were located in an area in the environment that did not provide them with the necessary nutrients to reproduce, or maybe strong winds came and damaged some of these plants. So whatever the event, okay, so here we're talking about unpredictable adverse condition or event. Okay, and now some of the plants are now destroyed, okay, by one way or another. Maybe an animal steps on some of these plants, another animal eats some of these plants, winds maybe damages certain of these plants, or some plants die because there's not enough water or enough nutrients. Okay, so whatever the case, what will happen is that we have only these, so these plants that I'm crossing may be damaged, and only 5 plants, these 5 plants, were able to reproduce, and they survived this unpredictable adverse condition or event. Now, only gametes from these plants will be used to make the second generation of the population. Now, the second generation of the population, the gene pool of the second generation contains more of the CW allele compared to the CR. So now, if we looked at the allele frequency here, we'll notice now the allele frequency of the CR allele decreased, while the allele frequency of the CW allele increased. Why? Because the plants from the first generation that survived the adverse condition or the basically the adverse event, they are heterozygous. They are mainly heterozygous, and one is, let's say, and one is homozygous Y. And these they were able to produce more of the gametes with more CW, let's say, allele, and this caused basically the increase in the proportion of the allele frequency of the CW allele. Okay, so notice here we have a decrease going from generation 1 to generation 1 to generation 2 in the frequency of CRW, and we have an increase in the allele frequency going from the CW allele frequency going from generation 1 to generation 2. Okay, imagine in generation 2 there is another basically adverse event, a second adverse event. Okay, that caused the damage into the population, so that only so that most of the plants within the population, again, maybe winds, maybe fire, maybe animals ate these plants, or insects damaged these plants, or maybe animals stepped on these plants, so that only 2 of this population survived. And as you can see, these two plants that survived the second adverse event affecting generation 2, these 2 plants, they have red flowers. So if these flowers, they started reproducing to produce the third generation, now all the third generation, or the population of the third generation, they all consist of red flowered plants, and all these red flowered plants, all the population contains only 1 allele. So the allele frequency here increased significantly, the allele frequency of the CR, and the allele frequency of CW dropped also, or disappeared completely from the population. So as you can see here, in one generation, the frequency of CR was high, in the next it's lower, in the next it's high. So this is what, so this is, if we model the CRW allele frequency, it was high, then it was low here, then it is high again. Okay, for the CR, for the CW, it was low, high in the second generation, then completely disappeared. Okay, so this is what is meant by genetic. This is an example of genetic drift that is caused by unpredictable events or unpredictable changes. So here we have unpredictable changes in the allele frequencies that are caused by random events or chance events. And these chance events or random effects, they affect the survival or the reproduction of individuals among the population. So we have two major effects that can cause the genetic drift. The first one is called the founder effect. This founder effect happens when a subset of the population or a few individuals from the population, they become isolated from a larger population. Okay. So imagine, let's say, we have a population of these red flowers. Okay. I'm just going to draw circles. And we have also pink. And we have white. So these are pink plants or pink flowers. And then we have white. Imagine if, let's say, by the activity of wind, or maybe by animals, or by whatever, a physical force, some of these plants or their gametes, okay, let's say wind or a storm, عاصفة, caused some of the plants to be blown from one location, from one geographical location into another. And the plants that were blown most by the storm or the wind, maybe. So here you have to understand the rest of the plants, they remain in their, let's say, geographical area, but some others, they might be blown away. Okay. So now we have a new population, a new smaller population is being developed. And now, if this population mainly consists of, let's say, mainly red and pink, let's say three flowers, one is pink and two is red. Okay. And now here we have a new population. Okay, this is the old population. So now you can see, if you calculate the allele frequency here, it will be different than the allele frequency in the new population. Of course, the plants in the new population, they will start reproducing and increasing in their number, and the frequency of the here, just without calculating, we could predict that the frequency of the CR allele will be much higher than the frequency of the CR in the original population. Okay. And the population, the CW allele frequency will be lower than the CW allele frequency in the original population. So this is the founder effect. This is one effect that will cause a genetic drift. Okay. Another effect is the bottleneck effect. And this happens when an event causes a very significant reduction in the population size. So you have a large population, and suddenly there's environmental changes, maybe a fire or a large storm or a very powerful storm that will cause a significant reduction or a significant decrease in the number of individuals within the population. Okay. So now, if you look at the gene pool, now the gene pool will not be reflective of the original gene pool. So the population, it's a large population, and now it becomes a smaller population. It is called the bottleneck effect because you have an original population, and This is the gene pool. Imagine we have 3 alleles, yellow, white, and blue. The yellow allele frequency is the lowest. Imagine if something happens, okay, an environmental change, let's say fire, and this fire burned most of the individuals within the population, and only few survive. The chances that the surviving population will consist of blue and white alleles, okay, will be higher than yellow. So it's called the bottleneck effect, because only few individuals will have enough time to escape, let's say, the environmental change. And these few individuals, they will continue basically living and reproducing. But now we had an original large size population, and now we have a smaller. And we have less genetic variation. The gene pool is dominated by the blue alleles. Okay, compared here, we have maybe allele frequencies that are very similar for the blue and the white alleles, and we have basically no yellow alleles, which means that the genetic diversity here, genetic diversity or genetic variation is less in the surviving population, the small-sized surviving population compared to the original population. Okay, so this is called the bottleneck effect. It's called the bottleneck because you have these balls representing alleles, and now they are basically should be poured into this cup, and let's say you have 5 seconds to pour as much as possible of these alleles into this cup. You can only pour, let's say, 6 alleles or 6 small balls, and these are mainly blue, and 1 is white, and there's no yellow alleles. Okay. Of course, human activities, industrialization, acceleration of industry and industrialization that was introduced by humans, and human activities in general, they could exert the bottleneck effect on certain species, plant and animal species, or maybe insect species in the environment. So we have certain, so human activities in general might affect other species, causing a change in the allele frequencies within the gene pool of these species. So in terms of the effect of genetic drift, so genetic drift in general, as a summary, so let's discuss, so example of genetic drifts, the founder effect and the bottleneck effect. So let's talk about the effects of genetic drift in general. Remember, genetic drift causes unpredictable fluctuation in the allele frequency. So genetic drift is significant and affects mainly small populations. So it's significant in small populations. The smaller the population, the more significant the effect of the genetic drift. Second, it can cause allele frequencies to change randomly. So if we go back to this example, so in one generation, high allele frequency of CR, in the next it's lower, in the next it's the dominant and it is fixed within the population. Here it's low, high, low. So this could happen randomly. Third, can lead to loss of genetic variation. Again, if we go here in this, so here we have red, white, pink in generation 1, red, white, pink in generation 2, only red flower plant. So here, because of genetic drift, there is no genetic variation in the third generation. Here, also in this example, we have 3 types of alleles that might be responsible for 3 different phenotypes. Because of the bottleneck effect, now we have less genetic variation. Still, we have some genetic variation, but it is the genetic variation here is reduced compared to in the surviving population compared to the original population. The fourth effect of genetic drift is that it can cause harmful alleles to become fixed. So there's a chance that, imagine if here the red allele or the CR allele is a harmful allele, and now it is fixed. It is the only allele found in the third generation in this example. Okay, so sometimes genetic drift might cause alleles that are slightly harmful to become fixed in a population. Okay, and this might pose a risk to the survival of the population as a whole. If this, let's say, if a harmful allele becomes fixed because of the genetic drift. Okay. So another process that can change allele frequencies is the gene flow. So in gene flow, alleles move between populations. So for example, if you have one population with a high allele frequency, let's say for the CR allele, okay? So this is one population. So population 1. And you have another population that is maybe far away from the first population, population 2. And population 2, the allele frequency of the R of the CR allele is lower compared to population 1. So you have mainly, let's say, pink and white flowers. Okay. So here you have again, you have an allele, let's say high allele frequency for the CR. Okay. Here we have a lower allele frequency for the CR. So imagine if these population or alleles moves between populations, because or gametes carrying alleles from both populations, they end up in the opposite population. So alleles or gametes maybe could be moved from this population to this population by insects, for example. So insects, they carry pollens. Pollens are حبوب لقاح. So pollens are actually, they contain gametes. So imagine if gametes from population 1 move into population 2, and also gametes moving in from population 2 to population 1. This means that there's a high probability that this population 2 will be receiving more CR alleles, and population 1 will be receiving more CW allele. Okay, so there's a large chance that the allele that is moving from population 1 to population 2 is a CR allele, and there's a big chance that the allele that is moving from population 2 to population 1, probably it will be the CW. So here we have low chance CW, low chance CR. So you could predict that after, you know, a couple of generations, these two populations, okay, eventually will have very similar allele frequencies of CW and CR. Okay, if this movement of gametes or individual plants, well, plants, they do not move, but if we're talking about animals, for example, also we could apply this to animals. If this movement of alleles or gametes or organisms or fertile organisms or individuals between the two populations, eventually the two populations will become very similar in terms of the allele frequencies of the CW and the CR alleles. So you could predict that gene flow actually tends to reduce variation among populations over time. Okay. And sometimes the movement could happen in one direction. So maybe this population, let's say, will lose CR alleles, okay? And this population receives the CR alleles, okay? So this population is losing CR, this one is receiving the CR, this one will, the genetic variation will change here, or sorry, the allele frequency will change here, the allele frequency will change here. Here, maybe the CW will be moving and genetic variation will be reduced. Here, maybe the CW is coming from this population. Let's say the CW is moving, and this means that also genetic variation is being reduced. Okay, so if CW alleles are moving, for example, genetic variation here will be reduced, and genetic variation here as well will be reduced. So there's an example in your book, just look at it, about snakes and how gene flow between two populations of snake might affect their genetic variation over time. So genetic variation, gene flow also can increase the fitness of the population. Fitness is basically the ability of the population, the capacity of the population to live in a certain environment, to basically to survive and reproduce effectively in a certain environment. So gene flow is known to increase this fitness of the population, especially if we're talking about exchange of alleles between populations made of individuals that they can mate with each other. Okay, just like in this example, we have two populations of wildflowers and individuals from both populations. They can interbred or they can mate with each other to produce fertile offspring. So if there is, for example, a missing allele that is important for the fitness of this population, it might receive that allele from another population, for example, from population 1, or which might basically increase the fitness. So for example, here, this population 2 has a low allele frequency of CR. Imagine if CR is important for the fitness of this population. So receiving a CR allele from the other population will increase the fitness of this population. So you could imagine here we have increased fitness because of just for example, because of the red or the CR allele, here we have reduced fitness. So receiving the CR allele from this population, from population 1, will increase the fitness of population 2. Again, fitness measures how well organisms are adapted to their environment, how well they can survive and reproduce in their environment. So gene flow eventually is also important in in the human populations, because now, because of innovations in transportation, now different or people from different countries, from different ethnic groups, from different continents, they can travel, they can migrate, and they can mate with people of other race, of other countries, other continent, or other ethnic groups. Okay. So before the development of fast transportation, let's say vehicles like airplanes or cars or trains, the rate of gene flow was, let's say, was low compared to nowadays. Nowadays, you could, in a couple of hours, you could move from one continent into another. Okay, and people migrated from different continents, from different countries, and now people of different race, of different ethnic group, of different, let's say, from different countries, they can mate, they can marry each other and produce offsprings. And this mixing between different race, different people from different countries, from different ethnic backgrounds, now increases change or causes a change in the allele frequencies in the human populations. This is the end of this chapter, and the next chapter, inshallah, we're going to learn about bacteria.